Hi, it's Dr. Bree coming to you from Hagrid's cabin, I guess his hut. Today we're going to cover molecular biology. I know that sounds scary, but we are only going to cover what we need to cover for us to have an understanding about the general building blocks of life and how they function together and how they stack on top of one another to get this like macro structure that looks like us. And then we'll cover Mendelian genetics. We'll also go a little bit beyond Mendelian genetics. We'll cross that bridge when we get there. But we'll look at Punnett squares and pedigrees. I'll walk you through some sample problems. All right, so starting with the molecules that are necessary for life, yes, it is important to the rest of the lesson that you understand these. So all organisms that are living on our planet, that's why they're called organisms, they are living. All of them are composed of four basic types of molecules that recur throughout all of those organisms. And these are essential for the cells, our cells that make up who we are, to be structured in a certain way to carry out their functions. So starting with proteins, proteins are basically amino acids that are strung together in long chains. They have these things connecting them called peptide bonds. That's not actually going to be that important for this particular class or this lecture, but just in case you're someone seeking additional knowledge, that is what they're connected by. So that you can envision them kind of like beads and they get put on a string. The string itself are those peptide bonds that space them out from one another and make sure they carry along all their buddies that are down the line on that string. Proteins interact with other proteins and other molecules that we will discuss here shortly. They are super crucial. I mean, even if you go to the gym, then you know that your muscles are made up of protein fibers. There are so many different kinds of proteins in your body that are doing all sorts of cellular tasks. Things like receiving signals from outside of the cells and saying, hey, this is what we need to do now, or this is the thing that we need to make. Sometimes they are the product, they are the thing that is made in response to a signal coming from outside the cell or within the cell. They can mobilize responses like calling, like inflammation, right? They can say, hey, there's a problem here that we need to work on. Immune system, please, like saddle up, come ride with us. So proteins definitely have a 3D structure, and I will get a little bit into how that 3D structure comes about, how it folds itself when I get to amino acids and their properties. But protein, so amino acids are the essential building blocks of proteins. Once the chain is complete, and again, we'll go over how that chain gets to the completion point. It's free floating and it folds into its 3D structure, and that is what we know as the protein itself. Number two, the next molecule that's necessary for life are things called lipids. These are fats, oils, and hormones. All of those things fall within this classification. Fats are the storage system for the body. So if you have a little extra on your body that you're not happy about, just know that you're storing energy that you can mobilize later. Now, hormones are a little bit different. Hormones are basically little chemical messengers in your body. They kind of take messages, I guess I would describe it as, between cells and tissues to get, they're running with the mail and they're trying to make sure that everybody's on the same page as what's going on right now. What is the stimulus that we're receiving from outside the body? Is it too hot, or are we supposed to be developing our secondary sex characteristics now? That's puberty. Puberty involves a lot of activation of hormones. So basically, your body's trying to read what's going on right now and what do our hormones need to do in response to that. When you are growing, you have growth hormone that's on active duty inside your body. It's like, hey, we are still building all of these materials, all these tissues in your body. We need to be in overdrive. But as you get older, those hormones start to settle down. Unless you have some sort of disorder, it is possible that like your pituitary still secretes growth hormone throughout your lifetime, and you can end up with something like gigantism. But your hormones, it's like a concert. They're listening to where you're at in your life and figuring out what should I be doing to respond to that. Now, lipids are very interesting in how they are I wanted to say designed, but that's not what I mean. They're the anatomy, the microanatomy of the lipid is very interesting. So what you're looking at in the image on the screen, you've got these little guys that to me look like the jellyfish from SpongeBob SquarePants, with only two legs instead of multiple legs. So they've got this real, like very round bulbous head, and then they've got these two little like dangling tentacles. And what makes up the outside of our cell walls, we are animals. I'm going to compare our cells to plant cells here in a second, but what makes up the outside of like animal cells is a double wall layer of these lipids. And you might wonder, like, how do they know to put their little tentacles in towards each other and put the heads on either side, on the outside part? And the cell doesn't know anything. It doesn't have a brain. It doesn't think. All of this has to do with chemical properties. So to break it down, that round head, like the top of what would be the jellyfish, if we're still going with the SpongeBob analogy here, it is hydrophilic. Again, let's break it down into the Latin pieces here. Hydro, think of hydro flask. What does it relate to? Water. Philic, I don't know if you ever heard like a bibliophile, or you can even think of this. I don't know if this is the same root here, but like phonetic, it's got the same phonetic thing going on, but that means somebody who's there's an attraction going on there. Like a bibliophile is somebody who loves books. So philic just means to be drawn to be passionate about, to be attracted to, that kind of thing. So in a hydrophilic head of a lipid is the piece of the little jellyfish that is attracted to water, has no problems with water. Now, the tails, these two little tentacles that dangle down, that form the inside portion of this cell membrane, they are hydrophobic. What's a phobia? A fear. It causes distance, right? So the little tail components here, they are in essence afraid of water, and we are made up of a lot of water. So the little tails, they can't be pointing towards the outside, the exterior of the cell, because the outside's made of water and they are afraid of water. That's their chemical property. They don't combine well with water. So to get an example in your head to visualize what this actually like might look like, think about if you put oil and water together and you mix it up. Do they actually mix all the way together where they make a new mixture where you can't see the oil and water separately? No, like it will for a second, but then when you let it settle, it'll settle into oil and water, right? Like you'll see two different layers. And this is because oils are lipids, very lipid heavy, and lipids are largely, most of the components of them are hydrophobic, meaning water repelling. So that is kind of what happens. That's how this membrane works to keep all the stuff in the cell and largely separated from the water. However, water can still permeate because this obviously is not a solid wall. So it's a really cool structure and mechanism, the biology of understanding how things move around in our body and how there doesn't have to be like conscious thought going on. The ways that these things have certain properties dictate a lot about how they organize themselves and how they function and relate to one another. All right, moving on to a more fun one for me, carbohydrates. Carbohydrates are basically sugar molecules, and they serve as energy for the cells. This cellular energy is called glucose. Now, if you are a runner or you are into nutrition and things like that, you might be aware that there are two basic umbrella categories of carbs. There are complex carbs and there are simple carbs. And this is because a carb is not a carb is not a carb. Like not all carbs are the same. Oops, sorry, hit a button there. So complex carbs tend to be more starchy. They tend to be harder for your body to break them down. So what this means is that because your body's working harder to break them down, you get a more prolonged release of that glucose, of that energy that's stored within those carbs. So we've got things on the screen like we've got fruits and veggies, we've got starches, like potatoes, tubers, things like that, whole wheat bread. There are tons of different kinds of complex carbs, but complex carbs, they they're the thing is like this, the starch is hard for our bodies to break down. So That takes us a long amount of time to break down, so very controlled release. So long-term energy, but a little amount of energy over that long term. That's complex carbs. Simple carbs are carbs that we can metabolize very quickly. So if you need energy, boom, bam, quick, these are good things to eat, but they are the things that are most delicious to us normally, right? Most of us like candy, we like soda, we like donuts, we like my dog's in the room, so I hesitate to say this word, but cookies. They know that one. That one they know. So things that are very, very easy to break down, a lot of fast foods. They are carbs, but because they're not so starchy and they tend to be like more processed. And processing is like pre-digestion. It's like already started to break down the components within this thing. So it's easier for us to break down those carbs, metabolize all that nutrition, not a lot of nutrition, a lot of glucose really quickly. So if you've ever heard somebody say, my kid has a sugar high or I got a sugar high, that's what happened. All that energy, all that glucose hit you at once. You hit this high and then you metabolize, you used it all and now it's gone and now you're on that low. And that's when it makes you feel real crappy, right? It makes you feel really bad. So complex carbs and simple carbs, they are the same type of molecule. They're made up of the same kind of thing, but because of the structure within those things, one of them gives you long-term small amounts of energy. So if you're going to do long-term running, like maybe you're going to run a marathon or something, that's probably a better way to go. But you might supplement with simple carbs, like little candies along the way to give you little extra bursts when you need it. But the problem is when you fall too much into, like, let's say the simple carb category, maybe and carbs, do not avoid all carbs. You need carbs. They're necessary for life, as you can see, but everything in moderation, right? If you fall heavily into the simple carb aspect, then that's going to wreak some havoc on your body because our bodies were not. A lot of our evolutionary history, we did not have a lot of simple carbs available to us, like these baked goods and these fast food things. So our bodies have not evolved to eat those things in high quantities, and it does have adverse consequences. Moving now finally to category 4, nucleic acids. These are the ones that you probably are familiar with. They carry the genetic information that is stored within our bodies and our cells, and not just us, other organisms too. And there are only two that belong in this category. That is DNA and RNA. DNA is deoxyribonucleic acid, and RNA is just ribonucleic acid. Um, they are very similar. They use a lot of the same bases. RNA is different in structure in that it's only ever exists as a single strand. DNA begins and most of the time exists as a double-stranded helix, but RNA is just one of those strands. We will cover, the purposes of why they look this way in a second and how they replicate and do the jobs that they need to do in the body. But the other difference I want to point out is that although both of them have 4 bases that they are comprised of, RNA does have one that is distinct from DNA. So DNA has, if you've learned the letters in the past, are A, T, G, and C. A is for adenine, T is for thymine or thymine. I've heard it both ways, but I think thymine is the most common. G for guanine and C for cytosine. So those 4 bases are the DNA bases, and they do pair with one another I'll go over them, but they do pair in regular fashion. Like you can predict what's going to pair with each one because they're very predictable. RNA has 3 of the same bases. It has adenine, it has guanine, and it has cytosine. But what RNA has lost is thymine, and it has replaced it with uracil. Now, out in the kingdom of living things, there are 2 main types of cells that comprise bodies or organisms' corporeal essence. Within our bodies, there are also 2 other types of cells. Those are the gametes and the somatic cells we covered in a different lecture. I'll come back to those. But just broadly speaking, in living organisms, there are 2 main types of cells. And the word cell does have, of course, a Latin root in it. It comes from cella. I don't know. I don't speak Latin, so I just imagined how someone would say that in Italian. And basically, it just means small room. And it's called small room because it has a bunch of things inside of it called organelles. And those organelles, imagine those are like the furniture in your house and the house walls itself or your room walls, that's That's what we're thinking. That's what the outside of the cell is like. So the two main cell types in all organisms are either, an organism is either a prokaryote or a eukaryote. It is one or the other. So prokaryotes are single-celled organisms, things like bacteria. And the key here, there are two key things that differentiate a prokaryote from a eukaryote. So a prokaryote doesn't have a nucleus. So, and a nucleus is, and I'll show you an image of it on the next slide, but oh, I've got one here. I forgot. I've got a nucleus is like this round sphere, and it contains genetic information in there. But the cell also does have other organelles, like little things that float around and have tiny little functions in there. They've got specialized tasks. Now, prokaryotic cell, because it doesn't have nucleus, its genetic information just free floating around in there. Its organelles, it's little things inside that have these specific tasks that they do, like creating energy, generating energy. Those in eukaryotes tend to have a little membrane package, a little envelope on the outside of them that keeps them self-contained. In a prokaryote, we don't have those. Those things are also just free floating. So imagine it's a prokaryotic cell is just like soup inside, kind of. So single-celled, doesn't have a nucleus, and all of its little organelles, so there's like mini versions of your organs, right? So same thing in a cell, in a microcellular way, and they're just free floating around in there. All right, moving now to eukaryotes. So we are eukaryotes. Our cells have a nucleus. They have membranes that surround their organelles. And eukaryotes can be single-celled organisms, or they can be multicellular organisms. We are multicellular organisms. But prokaryotes can only be unicellular, only a single-cell organism. So plants and animals are both eukaryotes, but our cell structures are quite different. I've got a picture of an animal cell on the screen. Animal cells we are animals, so we fall into this category. You can see that they are round and that they have that membrane that I just showed you a picture of what that actually looks like. Here it's just drawn as a line, but I showed you it's those lipids that surround the outside of the cell with those hydrophilic heads and those hydrophobic tails. So the animal cell is permeable, allows water to go and other things like calcium to go through channels. Plant cells, I am not a botanist. I don't really know that much about plant cell structure, but I do know a couple of key things that are important distinctions and contrast to the animal cell. So plant cells tend to be square, and they tend to be, the outside of the cell tends to be very, very thick. This outer membrane, so they have a membrane like us, this outer membrane is called cellulose, and it is definitely not as permeable as the animal cell membrane. And that's because plants really need to work hard to retain their water. Humans, we mobilize our water in different ways, right? So when it's hot, what does your body do? It sweats. So you need to be able to move water around in your body, inside and out of your cells. You need to be able to move from one part to another. So it is very important that we move water. Plant cells, it's not really the case. They don't really need to do it that way. And if you want an idea of like how firm cellulose can be, think of if you've ever eaten celery, you know, that crunch and it's got like all those fibers. Like imagine that's what the cell wall feels like. It is that kind of like firm. It's very structured. They stack really nicely on top of one another. To give that plant the very nice uniform structure that it needs. So the cellular structures that we're looking at, the organization patterns here, reflect how the organisms work biologically. One needs more structure to it to retain more water. That's plant cells. Animal cells needs to be a bit more dynamic and need to mobilize water a bit more. All right, so that's it for, okay, not totally it for the building blocks of life, but that's mostly it. So now we're going to kind of switch gears and talk about genetics. Genetics is the study of heredity. So we're going to look at hereditary patterns, how you predict them, some methods to analyzing them. And then we'll cover patterns, types of mutations, that sort of thing. In trying to decolonize the literature, one of the things that I like to always make sure to mention is that the very first image that we got of DNA's structure was in 1953, and it was brought to us and it was captured by Rosalind Franklin. I believe there was another intern there. But the reason I am specifically saying Rosalind Franklin's name is because you may have heard the two names Watson and Crick tied to the discovery of DNA. And in fact, they were the first ones, I believe, to describe DNA's double helix structure. So a two kind of think about a screw, the lines on a screw. But two of those intertwining. So if you can't see the image on screen, that's basically what I would imagine. Two curly cues or two ribbons that are intertwining to make a double ribbon. But Watson and Crick absolutely were part of this. But the reason that Rosalind Franklin's name is not one that comes to people's mind when they think about DNA is because she was a woman. And at this time, women were very much not respected in the scientific field. In fact, many women published under masculine names or their husbands' names because they knew they weren't going to be taken seriously in science. So I just want to give some of that power back because Rosalind Franklin, this is amazing to be the first person to get a picture, to get an image of what our genetic structure looks like is insane. And to have seen her left out of the narrative for so long, it explains why so many women feel that they can't be part of STEM. So anyway, a little bit of decolonizing stuff there. Okay, so DNA is deoxyribonucleic acid. Most people just call it DNA because who wants to say that long word all of the time? And it is made up of nucleotides, which we refer to as bases, and it has a sugar phosphate backbone. That is the thing that is running up along the outer edges of DNA, and then the bases pair up in between. I want to say in the middle portion here. All right, so this is not going to be super important for our class, but just so I'm doing my due diligence here, sending out to the world, DNA has the two strands have a organizational structure that's called anti-parallel. So basically, scientists call one end, they tag one end as what they call 5 prime, and then the other opposite end of that DNA strand they would call 3 prime. So that means that the paired strand with DNA, that other one, it runs parallel but anti, so it runs the other direction. So whereas the one on the screen, let me move my face because I'm in my own way. This one starts at 5 prime and runs to 3 prime. This other parallel strand starts at 3 prime and runs to 5 prime. So if you see 3 prime, 5 prime, it's just telling you that those strands are running, the start and end points are anti-parallel to one another. It's not going to matter for our class, but do what you will with that information out in the world. So there are 4 types of DNA nucleotides. I briefly mentioned them earlier. We're coming back to them now to make sure we solidify that knowledge. They are adenine, which we abbreviate as A, thymine, which we abbreviate as T, cytosine, which we abbreviate as C, and guanine, which we abbreviate as G. Sorry, I can't do 2 things at once. I forgot to keep counting. So each of these base pairs, and this is the way that they typically pair in the genetic code. A pairs with T, C pairs with G. They are paired by weak hydrogen bonds, and this has pros and cons. Now, for your DNA to replicate itself, it needs to split into this 2 different strands. So it needs to unzip itself basically in the center and split into 2 different strands. If those hydrogen bonds were so super strong, it can't do that. So you wouldn't be able to replicate your DNA and you wouldn't be here today. So it is helpful that they are paired by weak hydrogen bonds. However, because they're paired by weak hydrogen bonds, it means that mistakes can be made. So a couple of these bases are pretty similar in like polarity and charge to another base. And so it's not DNA, it's an enzyme, but we can accidentally get some mispairings that occur, but they stick because there's just enough of a weak bond between the two. So really it allows for mutations to happen. That can be a con, sure, but that can also be a pro because there can't be variation in life. There can't be improvement in fitness if there aren't mutations and variation for natural selection to work upon. All right, so just a brief practice here. If we've got a DNA strand that says that this is the order, this is the bases that I'm reading. I've got a C, A, T, G, C, T. I forgot my letters for a second. Okay, C, A, T, G, C, T. we are going to figure out what the anti-parallel strands sequence is. And so I will actually, this is the part I've been dreading. I'm going to have to write on the screen and I don't have a ThinkPad. All right, so C, you can look up here. C, cytosine always pairs with guanine, except in cases of mutation. So C pairs with G, so that will be the first letter that we start our chain with. Oh, I'm going to have to write way bigger than I typed. A pairs with T. T pairs with A. G pairs with C. C pairs with G. And T pairs with A. So if you got this problem on a quiz, test, homework, whatever, and they asked you what the complementary strand was, these are each of the base complements to the bases in this strand. Okay, I like to always start my practice problems in my lectures like simple and then work them towards the more complex ones so we can build on the information that we're gaining along the way. Okay, so this is how we work DNA complements. Okay. So I also think it's important for my students to understand like how crazy long DNA is because it helps us understand why it's packaged in a particular manner. So if we took the DNA out of a single cell in your body and we stretch it out end to end, you can take a guess in your brain as to how long you think that would be. It would be about 6.5 feet. I actually had a student guess a football field one time. I was like, dang, that would be even more impressive. But 6.5 feet is the general length that it would be. Now, not all DNA is the same length, but we're generalizing here. So if we've got 6.5, I guess you can't see more than that, 6.5 feet of genetic code end to end, and we've got to store it in a cell that I can't even see with my naked eye. We got to figure out some way to package that bad boy up so it's compressed, right? So we can fit it in all of these cells that exist in our body. So it's got to be very compactly organized. So what happens is here on the screen, I'm taking you from the microstructure up to the macrostructure. So first we've got our double helix. Now this starts to be wound around these things called nucleosomes, not relevant for our class. These things turn into you can see that they're all attached. It almost looks like beads on a string. So we're basically just winding DNA up on these nucleosomes and histones. Histones, you can see they're wound up in there too. So we're winding them up and we put them in this bead on a string structure. That thing is called a chromatin fiber. So we're starting to package it. If you can't see this on the screen, think of winding up a hose. If you wind up a hose outside, it's a lot like that. We're winding up the DNA around this round thing so we can store it more compactly. When we've got a bunch of those compact wound hoses, and in this case, the hose is the DNA, that is what we call chromatin fiber. Now, chromatin fiber gets long enough that we put it into yet another structure. So we compact it yet again. That is what we know as the chromosome. So these wound up chromatins turn into chromosomes. And I'll get to the structure of chromosome in a second. I don't want to jump the gun. So chromosomes, you're probably used to seeing them as the X structure. That is part of it. I mean, that is what it is, but I don't think people actually understand what that X is made up of. So one arm of that X is called a chromatid, So two of those put together and attached at the centromere, the little central meeting point of that X, they form what you know as a chromosome. But both arms, both chromatids, or both halves of the X, let's say, both halves of the X are identical genetic information. So all of the genetic information that you would need for that chromosome is on one singular chromatid, but it's duplicated on the other arm because the cell is going to have to divide and replicate itself. That's why we need duplicates. So really we're going to talk about just the chromatid, the singular part of the X, the singular half of the X right now, because the other part is just a duplicate. So most of our body cells typically have, and I say typically because mutations occur, things happen. Sometimes things don't split up correctly in the cell and we can have duplicates or we can be missing some. But most of our body cells have 23 pairs of chromosomes, meaning that we have 46 chromosomes total. And these are in the nucleus. So the number of chromosomes definitely vary by species. We've got 46 total in a cell. Chimps have 48, so they have a little bit more than us, and hermit crabs surprisingly have 254. The reason I'm pointing this out is not because I think it's cool, even though I do think it's cool. Um, it's because people tend to think that humans are so much more complex than any other creature. And what they don't understand is the amount of genes that you have doesn't necessarily correlate to complexity because a lot of what exists in your DNA codes for absolutely nothing. It doesn't like it might code for a defunct gene that hasn't been used in a while. It might be just remnants of a gene that we've lost. Sometimes it's just gibberish, like the DNA that's strung together. It doesn't create any particular protein. Um, so a lot of what is in your genome is what we refer to as junk DNA. Some people push back against that name because it conveys it like it's not helpful at all, or it wasn't even helpful at one point. And most of the genes that we have, I don't know actually most, but like if you think about how long evolutionary history has been, like at some point, I guess they probably could have done something. But a lot of it's basically gibberish. It's pieces of words strung together. Think of like minion speech, so some of it makes sense, but then in conjunction with the rest of it, maybe it doesn't make sense. So yeah, you can have a ton of genes and or a ton of chromosomes and not have a ton of active genes just because that's how the random nature of genetic coding worked out. So what you're looking at the screen again is a chromatid, a singular half of that X of the chromosome. We've got a short arm up top. We've got a long arm down bottom. We've got the centromeres, that central point where it would connect to the other chromatid arm. And there are two like I kind of call it caps basically on the end of the short arm and the bottom of the long arm. And those would exist on the other two too. So we've got these caps. I don't, that's just what makes sense to me. They're like little hats that go on the ends of these. And those are where a lot of junk DNA is stored. So like they still got bases and stuff, but they're not really coding for anything important. And the reason for this is that as your cells replicate and divide, those telomeres start to get eroded. So we think of the game telephone where you whisper in somebody's ear and then they whisper in somebody's ear and you're supposed to say the same thing all the way around, but then you couldn't quite hear what that person said. So the message changes along the way. So it's kind of like that, where the telomeres get ground down with a little bit of every iteration. And then eventually, when the telomere no longer is there, we start to get erosion of the actual important genes and genetic information that we do need. So those telomeres serve as like a protective buffer during cellular division. All right, so I talked about how DNA needs to replicate itself for you to be here today. That's absolutely true. So for life to continue, cells have to divide and replace themselves. And this relies on a process called DNA replication, which is where new DNA is copied from an original DNA template. And that's why we call the original strand the template strand. So DNA has three basic steps to it. The first step is called initiation. This is, if you know that word in general, you know it's the beginning. So this is when your DNA, your double helix shaped DNA, it gets unzipped. It gets split into two separate strands. This happens via an enzyme called helicase. Helicase comes in here and unzips the DNA. So we had a double helix. Helicase comes in, unzips it down the center, and now we've got two strands that were from that original piece, and both of those can serve to be template strands, and we can just attach new bases to this strand, and now we've got two new strands of DNA. I'm going to get into the rest of how that happens though. All right, Step two is elongation. So this is where we're going to assemble together some new matching DNA strands. These are called daughter strands from those two template strands that we had in our original double helix. So another enzyme comes in. Enzyme is called DNA polymerase. DNA polymerase comes in, and it is this yellow mouth-looking guy that's running down the DNA template strand. And it's basically reading. It's reading what bases are on that template strand, and it knows chemically, not consciously, right? It knows chemically what other nucleotide is supposed to pair with that template strand. So it says, I could feel that I read an adenine, so I need to add a thymine here. So it just puts it, and all of these nucleotides are called free nucleotides. Your body is really good at recycling things. So when strands are broken apart or cells are like die off, anything that can be reused will be, and you can also produce new ones of these too. But anyway, so you've got these free floating nucleotides out in the, let's just say cytoplasm, in the cell area. And polymerase is just saying, okay, this matches with this matches with this, and it's popping it on, just like Legos, okay? I read a C, I'm going to pop on a G. I read a G, I'm going to pop on a C. I read a T, I'm going to pop on an A. And you can see that it's creating a second strand that's going to, when released, have this double helix structure. Finally, DNA can't do that forever, right? DNA polymerase can't just keep adding bases forever. Otherwise, one strand would be too long. It would go longer than the other strand. So what it reads is a series of 3 DNA bases in a row. Those are called codons. So series of 3. So if you had like CAT, that's a codon. GCC, that's a codon. So it's just a series of 3 bases, and that codes for something. That's why they're called codons. Anyway, sometimes though a series of 3 bases screams the message, stop, release, you're done. And sometimes they scream start. But in this case, we're talking about being done. So they scream, stop, you ran, this is the end of the DNA chain. Stop doing your job, DNA polymerase. And so DNA polymerase is like, oh, dog, I got you. And it releases. It releases. It's done. It says, I will go find another cell that needs me to do that. So the end result of replication is going to be a cell with double the amount of chromosomes, so that now it can go about dividing its actual self into a new cell. Now, these are what I was talking about. These are codons. These are a series of 3 bases. This is a codon chart. This is how you would read what codon codes for. So if your first letter is C, we're going to be in this row. If your second letter is also C, then I know I'm going to be at least in this square. And then if my third letter is T, then I know I'm making, this is my codon, CCT, and that's going to make proline. That's the amino acid I would make. But you can see that some of these codons, some of these bases in a particular order code for stop. So TGA is stop. So at the end of the coding DNA, you would hear like stop. And polymerase would know that's when its job is done. So we're going to come back to this chart, but just to give you a sense of what codons look like and how they code, these are all amino acids on here. And that will be important for protein synthesis. All right, so I told you DNA is not always perfect. Like the replication process isn't great. So the ideal outcome for the cell is that it gets 2 identical strands of DNA, but mistakes happen. I mentioned those weak hydrogen bonds. So when that mistake happens, it's called a mutation. And mutations are the only source of brand new variation in any organism, which mutations can be bad, but mutations can definitely benefit us as well. There are a couple of umbrella ways to describe a mutation. They can be deleterious, which means harmful. They can be beneficial, or they can be neutral. Sometimes, so I'm going to go back one screen. You see how CCT and CCC and CCA and CCG all code for proline. That's the amino acid all of them code for. So that means if I have a mutation in this last letter, this third letter in the codon, I'm still going to get the exact same amino acid. So that would be a neutral mutation. It didn't change the outcome of the situation. So, but let's pick a different one. So like here, if I went from TTC, but I had a mutation here and now I end up with TTA, I'm coding for an entirely different amino acid. So that could have bigger consequences. So when we have something like this, where it's just the one letter that gets changed and no actual bigger outcome as a result of this, we call this kind of like a silent mutation or a synonymous mutation. It's making a synonym the same thing. So those tend to be neutral. All right, but let's look at a couple different kinds of mutations, classes, substitution, insertion, and deletion. Substitution is when one base is substituted for another accidentally, and that often happens with the bases that are very similar in chemical composition. So first three letters in our template strand here are CGA. Now, at the next iteration, something accidentally happened and we ended up with CCA. So our G has been substituted with a C. Oops, our bad. So that can be neutral. It could be harmful, but it probably is neutral most of the time. Now, let's just pretend this new strand is our template strand. So we've got CCA as our first codon. Now, here's where we've got insertion. We've got an extra base that accidentally got inserted into the strand. So if you count the number of bases here and the number of bases here, we've got one additional one now. So now instead of CCA, we have CCGA. And these ones are really harmful because they shift all the letters down the line. So where we started here, the first codon was CCA. Now I've got CCG. Let me get a pen here. My next codon, so here's codon 1, CCA. My next codon would have been ACT. And then if I look down now, what do I have? AAC. And then my next codon was going to be CGA. And now it's TCG. So it changes every codon down the line from that insertion. So odds are one of those is going to be really bad, right? One of those is going to be harmful. So insertions, really bad form of mutation. But same thing with deletion. So deletion is, oops, a base accidentally got left off. We didn't add it in, should have been in there. So here, now this is our original template strand. So we have CCG and CCG. Okay, nothing's changed there. That's fine. Then we had AAC, AAC. We've got, that's fine. But look, we've lost the T. The T was, accidentally deleted. So now instead of TCG, I've got CGA. So again, a deletion would change all the codons down the line, and we probably would have a problem there. So insertions and deletions tend to be the most harmful types of mutations because they change everything, all those codons that come after them. But substitutions, because they don't add or delete an additional base, that they tend to be more often than not like silent or neutral. All right. So I mentioned when we talked about eukaryotes and prokaryotes that when we got to our bodies, there are also two kinds of cells in there. So we're eukaryotes, but within our own bodies, we also have two other kinds of cells. Those are somatic cells, which are your body cells. Soma means body, but I tend to think of this somatic, like it has a long O sound and a T, and I think of toe and mitosis. All those things are giving me the same vibe, like toe, T. I don't know. Mitosis is like my toe, and my toe is made of toe cells, which are somatic cells. I don't know. Choose what makes sense to you, okay? I just try to give people ways to remember stuff. So somatic cells are your body cells. They want to replace themselves with identical things. If you are a cheek tissue cell, you don't want to be replacing yourself with a toe tissue cell because now you're going to have a toe coming out of your face. So you want to make, when you are a tissue cell, somatic body cell, you want to make identical copies of yourself to replace yourself so everything stays the same. So this type of replication that happens in body cells is called mitosis. So mitosis, mito cells, somatic cells. That's how it works for me. So these two daughter cells should technically be identical to the original cell here. Again, mutation can happen, but that is the ideal outcome. Two daughter cells that are identical to the beginning cell. Now, germ cells or gametes, those are the sperm and the egg. Now, these reproduce through what is called meiosis. No T there, meiosis. So this, in this one, you're going to end up with 4 daughter cells. So here in mitosis, we've got 2. In meiosis, we're going to end up with 4 daughter cells that are not identical to this beginning one up here. And that is important for genetic diversity. Genetic diversity is important for the longevity of our species. So what happens is if something in here called genetic recombination, where particular parts of the chromosome swap over, they switch, the genes get shuffled around so that we get multiple variations. And this is why even if you have a fraternal sibling that you don't look really the same because this all happens independently. Identical siblings are a whole other thing that happens through a different mechanism that is not within the scope of this lecture, but if you're interested, go look it up because it is interesting. But this genetic recombination is a step that does not happen in mitosis because mitosis wants duplicate cells, meiosis wants genetic diversity. So the cell cycle itself is pretty interesting. It is highly controlled. There's a series of checkpoints within it to make sure that the cell isn't doing anything aberrant. If it fails to pass a particular checkpoint, the process of, if that checkpoint is like something that can't be fixed, then the process of division, of cell division halts because everything's screaming, hey, we've got a problem, we've got a problem. So things that can cause the failure of a checkpoint are like DNA gets too degraded, like there's too many mutations, or maybe only half of it got coded, or a lack of nutrients to the cell, which means it's basically it's not a healthy cell. So those things can cause a cell to fail an important checkpoint. And if that happens, the cell can undergo what is called apoptosis. Apoptosis is really just cell death. It's a fancy word for cell death, but I like that it has this word pop in it because that is kind of what happens. It like the cell looks like it pops. There's a nuclear collapse, and then everything that can be reused or resorbed that was within that cell, all of the chemical constituents that can be reduced, reused, recycled by the body, they will be. So apoptosis is a natural part of the cell cycle. Cells cannot live forever because the longer they live and replicate, the more likely it is that they're going to develop mutations and they're going to go awry. All right, so we talked about DNA replication. Let's talk about protein synthesis. So proteins are a string of amino acids that fold up into complex 3D shapes. I mentioned that earlier in the lecture. There are 20 standard amino acids that can be strung together like those beads on a string to create proteins. And proteins have special jobs, right? Your muscle fibers, those are proteins that facilitate movement. Your muscles contract, and then it moves the bone along with it. So as you flex your bicep, it works with the bone. So muscle fibers facilitate movement in your body. You also have tendons and ligaments, but those serve more for stability than they do to initiate movement. Things that are other kinds of proteins are immunoglobulins, which help detect pathogens and mobilize an immune response. So when I talk about the 3D structure of proteins, we can look at, let's just concentrate, sorry, there's like a little dog hair on my nose. Let's concentrate on these charged ones. So if you've got an amino acid that is positively charged, do you think it's going to attract or repel an amino acid that is negatively charged? Think about magnets. What does positive attract? Negative, right? So those two are going to be drawn together. But what if you've got an amino acid that's positively charged and an amino acid that's positively charged? Those are going to repel each other. So that, just think of it that way. That's how the amino acid kind of gets, or how the protein gets its 3D structure, is that amino acids either want to be near each other or they don't, and it'll fold into the best position to meet all of those needs, all those different chemical properties and needs. So protein synthesis, your DNA contains instructions on how to make proteins. Proteins synthesis only has two major steps, DNA replication at 3. The first step of protein synthesis is called transcription. The purpose of this is to make ribonucleic acid. And basically we're translating, or we're transcribing, that's what we're doing. We're transcribing what we had in DNA. We're just copying it over via complements to an RNA copy. And RNA, remember, is single-stranded, has most of the same bases as DNA, except for thymine is replaced with uracil. The reason that we need to make an RNA copy is that DNA cannot leave the nucleus. It's stuck in there forever, and RNA can. And we need it to for this particular process. We need to be able to make the protein outside of the nucleus so it can be used for other things. So RNA is our vehicle to be able to get it to go do other things in other parts of the cell. So sorry, I gave you the answer to this, but uracil has been swapped, thymine has been swapped out for uracil. Okay, so we're still in transcription, right? We want to make an RNA version of our DNA template strand. So we've got the split here. We've got our DNA split up into two different strands. And now we have an enzyme come in that's going to read the DNA strand, the template strand, just like it did in DNA replication. In that process, DNA replication, we had DNA polymerase. Now we're making RNA, so we have RNA polymerase. So RNA polymerase comes in, it's going down the line, it's reading that DNA template strand, and it's attaching the RNA complements. So up here, where A would normally be, if we were making another DNA base, it would be paired up with a T. Since we're making RNA, we don't have a T, so we attach a uracil. So really, this would be the same process as creating the DNA template, except for any time you would want to put a T, you need to put a U instead. So we're going to practice this just like we did with DNA. So this is our original DNA strand, TAC, GGA, TGC. We're going to figure out our RNA complement. All right, so T pairs with what in RNA? It pairs with A. All right, so A pairs with what in RNA? Now, in DNA, it would pair with T, but in RNA, we don't have a T, so it would be a U. Then C pairs with what? A G. Then G pairs with C. Then G pairs with C. Then A pairs with what? Better not be saying T through the screen. It pairs with U because we are in RNA. T pairs with A. G pairs with C. And C pairs with G. Okay, so we went through the same process of how we would figure out the DNA ones, but every time we wanted to put a T, we said, no, 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 we're doing RNA. We're going to put in a U instead. So because this RNA's got to go about the world and do its things, and its only purposes in this specific instance is to help us create proteins. Oops. Because its only job right now is to help us facilitate protein synthesis, it wants to create basically a shorthand, like a shorter message. It doesn't want to carry around all those unnecessary codons that don't pertain to protein synthesis. So we've got our RNA bases here, but what happens is a process called splicing, where anything that doesn't really code for the proteins that we want to be making, the amino acids that are going to create the proteins, we're going to splice it out. We're going to cut it out, chop it up, cut it out, so that we have only the good stuff, only the stuff that's necessary to make the proteins in our little RNA. That is what's called mature RNA, and it is going to work on the step 2, which is translation. So translation is our second step here of protein synthesis. At this point, mRNA, M is short for messenger, so that's why that's why we call it messengers, because it can leave the nucleus, whereas the DNA cannot. So the messenger RNA leaves the nucleus, and it becomes bound to a ribosome. This ribosome is this big old blue sphere that's sitting on top of this mRNA, so they become bound together. The ribosome is going to go down that strand, that mRNA strand. It is going to read the codons there, the triplets, the 3 bases, that's codon. So it's going to read those, and each codon represents something. It represents either a start, a stop, or an amino acid. So this ribosome is going to translate our messenger RNA into a series of amino acids. The very first codon it reads should be a start. It should say, hey, ribosome, start your engines. You're going to start attaching amino acids now. So after that codon, it's going to read all the other codons. It's going to say, okay, this one codes for glycine. This one codes for cysteine. This one codes for aspartic acid. And it's going to use transfer RNA, tRNAs, are these little guys here, to bring that correct amino acid to attach to this chain. And it's going to pop it on, and eventually, when it reads the stop at the end of this process, it will release it, and this string of amino acids will fold into its protein structure. So I told you we're going to come back to these charts. Here they are. To figure out which amino acid is coded for, we are going to use our codon tables to figure it out. These are the 2 types of codon tables you might see. One is a square chart and one is a circular chart. I will show you how to use both. So let's imagine that our mRNA, our messenger RNA that our amino acid attaches to and is my dog is having a dream, and it's telling us which amino acids to attach. It reads our first codon is AUG, and we've got to figure out the amino acids that are coded for. So we've got AUG. So the way we would use this first table here, we would look at first letter. First letter is A, so I know I'm going to be in this row somewhere in this horizontal row. My second base is uracil, so my second letter is U, so I know I'm going to be in this square. And now my third letter is G right here. So I know that we're coding for methionine. Methionine is the chemical symbol for start. So technically that is a start codon. So the first amino acid we've got is methionine. Sorry, that's an E, methionine. Okay, now you can get the same thing over here by working inwards out. So your first letter is A. So we're going to be in this pie piece right here, this fourth. My next letter is U. There it is, U. And then finally the third letter is G. So that's going to put me in methionine again. Okay, so either way that you use the table, whichever one floats your boat, you'll get the same answer. All right, moving to our next template. The next strand is CCU. So CCU, first letter is C. I'm going to be in this row. Next letter is C, so I'm going to be in this one. And actually, it doesn't even matter what our third letter is because every single one of these makes proline. So this codes for proline. I'm going to do it over here. I know it might seem redundant, but I just like to give students the tools to choose whatever works best for them. So we've got C, C, and then C will put us anywhere here, so proline. So we get to the same thing. All right, finally, we've got ACG. So first letter A, we're going to be in this row. Second letter C, we're going to be here. And then third letter G. So we're going to make threonine. And the only reason I know that is because I do this a lot. So if I expected you to give this answer on a quiz or a test, I'd give you the full letter. Ain't no way I'm going to make anybody memorize all of these. Not for this class. So at the end of this, we're going to get one of these three stop codons. So if this chain kept going on, eventually we'd end in a stop codon. And at that point, the ribosome would release the chain and it would fold into its 3D structure based on those polarities and charges that we talked about earlier. All right, we are going to move now to Mendelian genetics. This is going to be a bit more interactive part. We already have talked about Mendel earlier in the brief history of evolutionary thought video. He was the monk who worked at the monastery and he was studying pea plants, and he observed that some parental traits seem to disappear in their offspring but then reappear in a later generation. And he used the concepts of dominant and recessive genes to describe, like recessive was used to describe the traits that would disappear and then maybe later come back, whereas dominant was used to describe the ones that showed up quite frequently. So he came up with these words to represent patterns that he was seeing in inheritance. And Mendelian genetics deal with traits that are controlled by a single gene. There are traits in your body, like your skin color and your hair color, that are controlled by more than one gene. Those go beyond Mendelian genetics. We are not dipping our toe in there right now. Mendelian genetics are traits that are controlled by a singular gene. I did briefly go over genotype and phenotype, but I'm going to hit it again here. So phenotype is the physical manifestation of a trait. So you get a combination of two alleles, one allele from mom, one allele for dad, whichever one gets expressed, whichever version of the allele gets expressed, that is phenotype. So things like pea pod shape, pea pod color, flower color, that kind of thing. Genotype is the word used to describe the two alleles that you get, the two copies. It's dominant, recessive, dominant, dominant, recessive, recessive, whatever. Those gene copies are called alleles, and your phenotype is determined by your genotype. So genotype is the letters, phenotype is the manifestation. I promise you'll have this by the end of this because I'm about to say those words about 1,000,000 times. So we use something called Punnett squares as a method to be able to predict genotypes and phenotypes of offspring of particular parental interminglings. interbreeding aspects. So if purple flowers are dominant, so here's just an easy question to start with. Don't worry, I'm going to give you a chance to do this on your own too. So if purple flowers are dominant, we would put the big letter. So you could use capital B if you want, or you can use capital P. You can use capital whatever letter. Most of the time, we encourage you to use letters that pertain to whatever the trait is being talked about. So if you're working a problem about freckles, you probably want to use F as your letter, but technically you could use any letter. Just needs to be a capital and a lowercase. So if purple flowers are dominant in our situation, we represent that with a capital letter. What is the possibility or the probability that two heterozygous parents produce an offspring with white flowers and an offspring with purple flowers? So we're going to look at a filled out Punnett square so that we know what to do on our own. First things first, you draw the square. But the square is going to have 4 squares on the inside, and you're going to have to put 4 letters on the outside. The two letters on the top represent one of the parents, and the 2 letters along the left side represent the other parents. Both of those will represent alleles. Now here they've got mom on the left-hand side and dad on the top side. It doesn't matter. You can put whatever parent, wherever. You're going to get the same answers. Okay, so we know how Punnett square looks. Now we're going to figure out parental genotypes. Genotypes. So we know that both parents are heterozygous. Prefix of hetero means different. So we know each parent is going to have one dominant allele and one recessive allele. So we put those along the outsides of the Punnett square. Now, to calculate the offspring probabilities in the inside of the square, we're going to do I describe it as cross multiplication, but we're just going to bring down and across certain letters. So for instance, this yellow square that we're starting with. On the top of that square, there is a capital B. So I'm just going to bring that right down into the offspring square. On the left-hand side, the first letter is a B, a capital B. Sorry, let me specify. It's a capital B. So we're going to bring that right into that square. That means that that particular offspring outcome, if each parent gave a capital B, a dominant allele, that offspring would be homozygous dominant, 2 capital Bs. Okay, moving to the square next to it here, we've got from dad, he's giving us a lowercase b, a recessive allele, and mom is still giving a dominant allele. So in that particular case, if that were the outcome, the offspring would be heterozygous. They would have one dominant allele and one recessive allele. Moving now down to this square on the bottom left. In this case, dad is giving a dominant allele and mom is giving a recessive allele. So this offspring would be heterozygous. Finally, we've got this last square in the bottom right. In this case, dad's giving a lowercase b, so recessive allele, and mom is giving a lowercase b. And our offspring would be homozygous recessive. Okay, that is how we filled out that chart. I'll walk you through it again. Don't worry, you'll see it a second time. It's a little bit harder when it's filled out here. But let's clear this. And now we're going to figure out our probabilities. So because B, the capital letter, codes for a dominant allele, anywhere you see that capital letter, that is going to be expressed in the phenotype, the actual manifestation. So in this instance here, I've got a homozygous dominant individual. They only have dominant alleles. That means they can only express the dominant condition, which is what? Purple flowers. So their phenotype is a purple flower. My heterozygous individuals, I got 2 of them. But I told you, anywhere you see a capital letter, anywhere you see a dominant, that's getting expressed. So both of these guys, even though they do have a recessive allele, they say, shh, recessive, you're not going to say anything. And the purple allele is expressed. Now our 4th offspring possibility here is homozygous recessive. It's got 2 lowercase b's. In this case, there is no purple allele to express. So finally, the recessive condition gets to live its best life, and we get white flowers. All right, so that's how we get to the phenotypes of each of those. Now, calculate some probabilities here. So what is the probability of an offspring with white flowers? So we're going to look at the inside squares here. How many of those offspring have white flowers? 1 square. One square shows white flowers, right? So we've got 1, and you're going to divide it by how many squares there are inside. 4. There are 4 squares. So 1 out of 4 offspring is that's the probability, should have white flowers. What is that as a percent? 25%. If you don't know how to calculate percent, it is 1 divided by 4 is going to give you 0.25. 0.25 times 100 will turn it into a percent, or 25%. If it helps, since all of these Punnett squares are going to be just using 4 individuals, you can think about how many quarters go in a dollar. So there are 4 quarters in a dollar. If you have one of those guys is white flower and these guys are purple flowers, you've got 25 cents. That is a white flower, or 25%. These guys, these purple guys, there are 3 of them, or 75 cents, or 75%. So the offspring with purple flowers, there are 3 out of 4 in that Punnett square, and that gives us 75%. Now here's a note, is that these 2 percentages, or the percentages of any genotype and phenotype, they should add up to 100. They should add up to 100%. If you're getting less than 100% or more than 100%, you have done something wrong. So that's a good way to just quickly check and make sure your math is mathing. See, I've got a blank one here so we can work it at the same time. So I'm going to read the problem, and then if you want to work it yourself first, I would pause the video, work it yourself, and then I'm going to walk you through it. So in humans, being a tongue roller, which is this, that's dominant over not being able to roll your tongue. And this problem has already defined the letters, so you should definitely be using those particular letters in your response. A man who is a non-roller, so he can't roll his tongue, marries a woman who is heterozygous for tongue rolling. Create a Punnett square and tell me the probabilities of a couple having a non-roller versus a roller offspring. Okay, so at this point, feel free to pause and work this problem. All right, so the first thing I'm going to walk you through is how do we determine the parents' genotypes? And that is what is going to go on the outside of this square. Remember, you can put mom on one side and dad on the other. It does not matter which side, you are still going to get the same answers. So dealer's choice. So being a tongue roller is dominant over being a non-roller, okay? We're going to look at the rest of this problem, which tells us something about parental genotypes. We know that a man who cannot roll his tongue marries a woman who is heterozygous. I'm going to take that one first because I feel like heterozygous is always the easiest one because it tells me 2 different letters, so I don't even have to think. I know it's what it's going to be. So mom, I'm just going to put her down left-hand side. She's heterozygous. I know she has 1 capital letter, 1 lowercase letter, 1 dominant, 1 recessive. That's what heterozygous means. So we're going to put her along the left-hand side here. Okay, moving to dad now. Dad is a non-roller. Okay, and what I like to have my students do who are just starting to learn how to do these problems is write out the possible genotypes. So R, and then little r. And define what that would look like as a phenotype. Okay, so for instance, we know that capital R, the dominant gene, the dominant allele, that codes for being a tongue roller. And if I have even 1 capital letter, even 1 dominant allele, I'm going to express that dominant trait. So what that means is that these have capital R's and there is 1 here. So both of those are going to express rolling. Rolling. What does that mean? So that means that over here, with this lowercase, or homozygous recessive, they don't have a dominant allele, so they can't express tongue rolling. So this one over to the right is the only combination that's going to give us non-rolling as an option. So that tells me, since dad can't roll his tongue, he must not have a dominant allele at all. He must be this one right here. So we've got homozygous recessive for dad. Now we're going to fill out our Punnett square. If you just needed a hint on the parents, pause here, do the inside of your Punnett square. Okay, I'm going to go over the inside of the Punnett square now. This is like cross multiplying. So this large R for this row, it's going to be carried over for this individual and for this individual. this lowercase r right here for mom, because she's heterozygous, that's going to be carried over to each of the Punnett squares to the right. Now we can look at dad. You'll notice I'm working this problem a little bit differently than I did the last one. Again, I like to show a couple different methods to show that you can get the same answer and you can just use what works for best for you. So now we're moving to dad. He only has lowercase r's to give. He only has the recessive allele. He doesn't have a dominant allele. Mom was rows, dad's columns. Okay, so our lowercase r is going to come down in columns here. Again, the only allele that dad can give is a lowercase r, is a recessive allele. So even if you weren't doing this cross multiplication type thing, you would know that you could put a little r in every single box. Okay, those are genotypes. We're done with genotypes. We filled it out. Yay for us. We're perfect. Now we're going to figure out what the heck those phenotypes were. And this is why I defined them at the beginning, because now it's really easy. So now I can be like, okay, I knew that heterozygous would be rolling. So this one can roll its tongue. This offspring would be, I should have just typed it again. Oh well. So these two will be rollers. And even if I didn't define it, I would know that if there is a capital letter, if there's a dominant allele, it will be expressed and the recessive one will be masked. So I would know that these two individuals would express the dominant trait, which is rolling one's tongue. This means that these two, they can't roll their tongues. Okay, so let's figure out the probabilities of these couples' offspring. So non-roller makes up, so that's these two squares down at the bottom, that makes up 2 of the 4 squares. What is that as a percent? 50%, right? So 50 plus 50 is 100. So we know, but we can check. So there are 2 boxes out of the 4. Again, that is 50. So it is a 50-50 chance for them whether they're going to have a kid that can roll their tongue or a kid that can't roll their tongue. Now, I don't know why you use a Punnett square for this if you were having a baby and you wanted to know about this, but normally we use it for traits that are more influential and matter more than tongue rolling, but you can. All right, so now we're going to level up and we're going to talk about a couple of other different kinds of inheritance. We're going to talk about blood type. Now, blood cells have antigens, which are proteins on their surface, and that's what codes for a particular blood type. Since there are 3 different alleles for blood, A, B, and O, we're going to have to do something a little bit more complicated here. So in this instance, O is the only allele that is recessive. A is not recessive, B is not recessive, O is recessive, meaning that in order to express O-type blood, you need 2 alleles for O. That is the only way that somebody gets O-type blood. So to be type O, you need to get an allele for O from both of your parents. 1 allele for O, 1 allele for O. That is the only way you get type O blood. So what does that mean for the other ones? For A and O, if they're not recessive and there's 2 of them, what does that mean? Well, it means that A and B are dominant together, which is what we call codominant. They are both expressed. So you can see A and B, both the A trait is expressed and the B trait is expressed. So they are codominant. So if your genotype is AO, if you've got an allele from mom for blood type A and you've got an allele for dad for blood type O, what is your phenotype? What is your actual type of blood? Well, O is recessive, so you would be type A. What about for BO? I just told you that B is dominant and O is recessive. What does that mean your actual phenotype is? B. B, because O is recessive. What about AB? Your phenotype would be AB because they are dominant together. So they are both expressed together. So this is called codominance. There is another type of weird dominance patterns where these would be blended. That is not this. In codominance, the traits are expressed separately, but in the same organism. So it'd be like if you had a dog that has white spots and black spots and it was a codominant trait, then they don't blend, right? They don't blend into gray. That's what it's like. So now we're going to work a Punnett square problem with the blood typing. We're going to create a Punnett square for a person who has O-type blood and a person with AB-type blood, and we're going to figure out probabilities for the offspring. So you can pause if you want to work this. So I always start, if there's a person with AB, I always start with them because it's the easiest one. It's like being heterozygous. And then we have another parent who is O type. To be O, I just emphasized it. But what does your genotype have to be since it's the recessive? It has to be O, O. That is the only way you get O type blood. I will keep saying that again and again until you remember it. Okay, so now we're going to do our crosses here, right? So O is the only allele that can go in this. However, O is recessive, so I'm going to write it to the right because you always put the recessive one, you always put it after the dominant one. So the only allele this person could give is an O, and the only allele they can give down here is also an O. Okay, now we're going to look at this individual. So we're looking at columns. This person is going to contribute an A down this column here, this first column. So AO, AO. Now the second allele here is B, so B is going to go down the second column, BO, BO. Okay, those are genotypes. I always try to get you, and I will put questions on the test that are like, put answers where what are the I ask you for the phenotypes, but I give the genotypes as a potential response to make sure you know the difference. These are genotypes. Those are the combination of alleles that you could possibly have in offspring. But what are the phenotypes? So this person here who has AO, what type of blood are they going to actually express? Well, O is recessive, so they're going to be type A. O is recessive here, so they're going to be type A. Okay, what about over here? These two offspring. What is their phenotype going to be? Not their genotype, but their phenotype. O is recessive, so forget that guy. B is dominant, so we're going to get B. O is recessive, so we're going to get B. So this is the same kind of situation that we had on the last example where it's a 50-50. So there's 50% chance that these individuals will have a type A, a child with type A blood, and a 50% that they would have an individual with type B blood. So if they have an offspring that's O, call Jerry Springer, because some happened there. All right, moving now to the next level up, which are pedigree diagrams. Pedigrees don't show you the specific genotypes. What they show you are phenotypes. In pedigree charts or pedigree diagrams, kind of same thing, males are represented as squares and females are represented as circles. This has to do with sex, not gender. However, it is also possible, sex is a spectrum, and I will cover that in a different lecture, but it is possible to be like XXY or XXX or XYY. So this is not as cut and dry. This is okay. This is not as cut and dry as the chart makes it seem. So when a square or circle is shaded in, like it's gray, and the other ones are like white, the shaded in ones mean that the person is expressing whatever trait that we are focusing on in the pedigree chart. It could be a recessive trait, it could be a dominant trait. It is whatever trait of focus is specified in the problem. So for a recessive condition, sometimes people, so people can be heterozygous, right? Like they can have that allele and not express it. In that particular circumstance, they would be called a carrier for a disease. So if a disease is recessive and you have an allele for it, but you have a dominant and healthy allele that masks it, you are a carrier. So this particular pedigree tracks individuals affected by Huntington's disease. Huntington's, what it is not relevant to this particular problem, but you can go look it up if you want. But Huntington's is a dominant Mendelian trait. You always want to read your question thoroughly to figure out if the trait of focus that everyone who has it, right, is shaded in on the chart. You need to know from the get-go, is that a dominant trait or is that a recessive trait? Because I promise you it helps. So we know that Huntington's is dominant, which means you only need how many copies of a Huntington's allele to express it? One. Because if you have one dominant allele, it masks the recessive. So what this means is that, here's our three combos. Remember, In a Mendelian situation, you can only have 3 potential combos. Okay, so Huntington's is a dominant trait. So if I have one capital letter at all, this person will have Huntington's. So this codes for Huntington's. I don't know why I feel like I spelled that wrong. I feel like it's the font. Huntington's. Okay, this person over here, this combo is homozygous recessive. No, dominant alleles are there, so they will not express it. So again, you don't have to do this. Once you get good at these, you can skip the like intermediate steps, but I try to teach for all levels. So I really just try to teach from the base and then you take what you need and you can create shortcuts when you need them. All right, so those are the possible combos, the genotypes that any of these individuals on this chart can have. So our goal is here, determine the genotype of the circled individual, which is up top. It's a shaded in square. All right, so how do I solve this? Okay, well, they're shaded in, which means what? It means they have Huntington's because shaded means they're expressing the trait of focus. Huntington's is the trait of focus. That means that looking up here at what I defined, this individual who's in the circle is either big H, homozygous dominant, or heterozygous. One of those two combos has to be true. So we've already eliminated one. Now, I don't know if you like to, but I find it very helpful to go through and write the genotype on top of these that I am certain about. So for instance, anybody who doesn't have Huntington's can only have one potential genotype. There's only one genotype that codes for non-Huntington's, and that's homozygous recessive. So little h. So I'm just gonna pop that in there. Okay, and so on and so forth. At this point, since I'm concerned with the top individual, it doesn't really matter past this generation here. But I do it for funsies. All right, so you could keep filling it in the rest of the generations. It's not going to be of consequence to us currently. All right, so we've got to figure out which of these two potential options makes the most sense. So what we do is we create a Punnett chart or Punnett square for both options. So that's a way big Punnett square there. For both potentials. Now mom is circle, remember? So because we have curves, that's how I always tell my students to remember it. Females have curves, so we're the circle. Okay, so mom, we know for certain because she doesn't have Huntington's that she's going to be homozygous recessive. So I'm just going to put my two little H's on the top of these two potential Punnett squares since I am 100% sure about her genotype. Now, on the sides of these Punnett squares, I'm going to put the two potential options. So for my first Punnett square, I'm going to put the homozygous dominant genotype, so the two big H's. And in this second one, I'm going to put the heterozygous potential, so capital H, lowercase h. And now, it's at this point in my lecture that normally students are like, well, I can already see the answer. That's great. Then you don't have to finish it. Like if you can already tell the answer from this, but some people can't. So we're going to work the rest of it. So here I'm going to fill in the middle of my Punnett squares. So I've got HH. So I've got that one's heterozygous. That one's heterozygous. That one's heterozygous. And that one's heterozygous. Now over here, we've got heterozygous, homozygous recessive, heterozygous, and homozygous recessive. And if you can't read my writing, I apologize. Hopefully your writing is better. I'm doing it on a screen with a mouse pad, so my bad. Okay, now we can take a gander at this and figure out, all right, let's look at this first Punnett square where dad is homozygous dominant and mom is homozygous recessive. Now we look at what are the genotypes here. Every single one, 100% of them are heterozygous. Now, what does that mean for their phenotype? Well, we can look up here, or we can come back here, and we know that if there is one singular dominant gene in that genotype, that they're going to express that dominant trait, which is Huntington's. So what does that tell us? It tells us that every single offspring in this particular scenario would have Huntington's disease. Is that what we see in the pedigree diagram? Does every single offspring, so all these lines that come down from them here, that all connect back here, those are all offspring. genetic offspring. And then this is like their wife or their mating. It doesn't obviously have to be marriage. You can have a kid outside of marriage. I'm raising my hand here. That's me. But those are just like reproductive interactions. So every single kid, every single line that's coming directly down from this breeding relationship, they should be filled in. They should be gray. If this was true, every single one of these offspring would have Huntington's. They don't. We see two individuals here have escaped it. They've come out without Huntington's. That means that this combination, homozygous dominant, cannot be possible. It's not possible with the situation that occurred. It has to be heterozygous. So this individual's dad's genotype is heterozygous. So let's try a different one. In this particular instance, we are going to track cystic fibrosis, horrible condition. So cystic fibrosis, I don't want red, let's start with blue. Okay, cystic fibrosis is a recessive condition. And this is where, again, I'm going to go up to the top and I'm going to write each of my my possibilities here. Okay, so now I got all my possible genotypes that could ever occur here in my Mendelian trait of cystic fibrosis, and now I'm going to write what the phenotypes are. So for cystic fibrosis is a recessive condition, meaning it's carried on the recessive allele or lowercase letter, if that helps you. So in this first instance, I have no lowercase letters at all, so this person cannot have cystic fibrosis. Okay, in the middle, in heterozygous, I do have an allele for cystic fibrosis. However, it's on the recessive allele, which means that the dominant one, the healthy allele, will mask it, and we will not have cystic fibrosis. This individual only has recessive, it only has recessive alleles, so there is no healthy allele there to mask it. So this person, this combo would have cystic fibrosis. Okay, so now we can go ahead and figure out, this is a two-stepper. We have to figure out the genotypes of individual 1 and individual 2. So individual 1, I think, is easier because I'll explain why. So individual 1 is shaded in, which means they're expressing the trait that we're tracking in this pedigree chart, which is cystic fibrosis. So that means they have cystic fibrosis. Which of our options was the only option that gives somebody cystic fibrosis? Homozygous recessive, right? So little c, that's what individual 1 is. All right, up to individual 2. We know that individual 2 doesn't have cystic fibrosis. They're not shaded in. So again, we have the same possibility. So we've got CC as a possibility, big C, or big C, little c. So it's going to be one of these. It's going to be heterozygous or it's going to be homozygous dominant. And they are mating with someone who has the same thing. Okay? We don't know their genotype either. So they could also be heterozygous or they could be homozygous dominant. So that's a little added layer of complexity here. Now I notice this individual right here. What do we know about that? That individual, number 1, we know that that's an offspring, a genetic offspring of that pairing. Number 2, what else do we know? They're shaded in. What does that mean? It means that they have cystic fibrosis. So their genotype is what? Little c. Ooh, that's hard to read. I'll write it to the side. Little c, so homozygous recessive. So what does that mean? If you're not seeing it yet, hang on. I'll draw the Punnett square. I like these because they're like logic problems. I'm very much into logic problems. Okay, so if this individual's genotype is homozygous recessive, that means that they got a recessive allele from mom and a recessive allele from dad. So that means both mom and dad had to have a little c. But mom and dad didn't have cystic fibrosis. So there is no way they can be this genotype, homozygous recessive, or they would have cystic fibrosis, which means for them to be healthy and not have cystic fibrosis, they need a dominant allele. They need a big C. This is the only combo. You can try it. You can write out all the possible Punnett squares. This is the only combo where both parents are heterozygous that will give you an outcome that will allow you to have an offspring of that relationship that is homozygous recessive, that has two little c's and has cystic fibrosis. So both parents, so the individual 2 has to be heterozygous, and it didn't ask me for it, but this individual that they interbred with is also heterozygous. So 2 heterozygous individuals. That is the only scenario in which this individual can inherit a little c from mom and a little c from dad. That's the only one. You want to level up pedigrees? Let's do it. This is the hardest problem I could probably give. It is about X-linked conditions. It adds another layer where you have to think about sex chromosomes too. So X-linked conditions are specifically carried on the X chromosome, and males tend to be more susceptible to them. Why? Because males only have one X chromosome. So if they, and I'm just going to say typically, because again, it is possible that you can have different combos of alleles. But typically, a stereotypical male will have only one X chromosome. Males often are X. Why? Oops. And females tend to be X. X. Okay, so if you're a male and you've inherited a mutation on that X chromosome, you don't have another X that could potentially have a masking dominant allele on it to help you. But if you're a female, it's possible you get one healthy X and one mutated X, and that healthy X can mask the recessive one and keep you from expressing whatever trait that is. So males tend to be more susceptible, particularly to things like color blindness, because they are attached to the X chromosome. So we're going to look at a pedigree chart that tracks an X-linked condition, which is Duchenne muscular dystrophy, or DMD. And we're going to work this problem exactly the same as we would have worked the other 2 problems, where the first thing I'm going to do is look and see what kind of condition I've got that I'm tracking in my pedigree chart. And in this one, we are tracking a recessive condition, So we're going to use big D and little d. Okay, so big D and little d. So I'm going to define my 3 possible combos. I'm not worrying about sex chromosomes yet. I didn't forget. Don't worry about them yet. Okay, returning back to the problem, we've got a recessive condition, meaning it's carried on the recessive or lowercase allele. First situation here, homozygous dominant, not possible, sorry. So no DMD there. This individual in the center, they have a dominant allele that is healthy that will mask the DMD recessive allele. So we've got no DMD there. Now, this last one, homozygous recessive, is the only one that can give us the expression of Duchenne muscular dystrophy. All right, now we can start talking about sex chromosomes in the pedigree chart over here. So we're going to look at person 1 and person 2 because I wrote this, because I know this problem. I know that person 2 is going to be easier, so let's work that one first. So person 2 is a square. Does that mean they are a male or a female? A male. So that means that their sex chromosomes would typically be X, Y. Okay, we're going to start there. Now, they are shaded in, which means they are expressing or not expressing the trait. They are expressing it. So since they're shaded in, we know they're expressing Duchenne muscular dystrophy. I'm just going to say DMD. It's easier. So they are expressing DMD. DMD is what kind of trait? Which allele is it carried on? Recessive. And it's carried on the X. So this individual is a male, so they only have one X chromosome. And they're expressing a recessive condition, meaning the only possibility here is for them to have the recessive X allele. Okay? If they had this combo, they would not have DMD because this is the healthy allele. This is the non-DMD allele. And it's the only one they can express because they only have one X. So they would, if this was their combo, they would not be shaded in. So it cannot be this. Oops, I just want an eraser. So let's get some of my stuff out the way. Okay, so person 2, I've already finished with that. This is person 2 right here. That's their genotype. Boom, bam, done. Halfway. All right, we're going to go over to person 1 now. Person 1 is a circle, which means they are a male or a female. A female. And females, typically their sex chromosomes are what? XX. Beautiful. All right, this person is shaded in. So they are expressing or not expressing the trait. They are expressing. They are expressing. Okay, now, because it's a female, we can have two alleles. Remember, so we got two X's, each X carries this allele. So these are the possibilities. So we could have this. Now, would this cause this female to express muscular dystrophy? No, right? Because these are two healthy alleles. The capital D is the non-mutant form, so you wouldn't have muscular dystrophy. So what if we had heterozygous? What if we had this combo? Well, we still have one of those healthy dominant alleles, so it's going to mask the recessive DMD allele. So that person still wouldn't have it either. And then as you can see up here, only combo that's going to get our person to express DMD is a double lowercase d, so a double recessive. So this is the genotype of this person, number 1. So let's go a little bit beyond Mendelian inheritance, but not far. I mentioned that there is another kind of dominance that is a little bit different than codominance, and that is incomplete dominance. This is when the heterozygous condition produces a phenotype that's a blend of the alleles. So basically, what we would consider the dominant allele, the capital letter, it cannot fully dominate the recessive allele, so they blend together. Now, if you watched my the previous video on the brief history of evolutionary thought, then you know that blending inheritance is what we thought was, in Darwin's time, that's what we thought inheritance patterns look like. And I said it was wrong. Well, not quite. Sometimes it can be like that. It's definitely much rarer, but so one example is snapdragon flowers. So the red color is the dominant allele, so we do write it with a capital R, but it is not completely dominant over the recessive condition, which are white flowers. So what this means is if somebody is homozygous dominant, if a flower is homozygous dominant, it has it's red in color. If a plant is homozygous recessive, it is white in color. But the heterozygous is where we see the difference here. So heterozygous means that there's a blend of those two characteristics. So the big R codes for red flowers, the little R codes for white. So the heterozygous condition, the outcome, the phenotype is going to be pink flowers, a blend, a nice blend. All right, so we've got a red snapdragon and a white snapdragon, and they are going to be crossed, and we are going to figure out the probabilities. So remember, red is our dominant, and white, little r, is our one for white flowers. All right. So for this one, we know that a white snapdragon has to be lowercase r, and the reason I know that, the annotations are still going to be on the screen, but don't freak out, is because I told you here. So a homozygous recessive is white flowers, and homozygous dominant is red. So I already know the genotypes there. So we know one of these is this, and one of these is this. Okay, so I know that this individual is going to bring only, actually, let me go the other way. You should start with the dominant ones. Okay, so I know that going down the column, we're going to have a capital R. Going down this column, I'm also going to have a capital R because this plant, that's all it can give is dominant alleles. This plant can only give recessive alleles, so all the way around the board, we are going to get a recessive allele. So that means 100% of the offspring are heterozygous. They are heterozygous. And what did I tell you about incomplete dominance in the heterozygous condition? What does that mean? That the phenotype is a blend. So the blend between red and white is pink. So pink flowers in the offspring, for all of these heterozygous, every single one's going to be pink. So that answer is going to be 100%. Now, without even looking at the other ones, we know those, without even looking back to my Punnett square, we know that the other two answers have to be 0, right? Because I can't have a number that if I add all these up, they go above 100% or below 100%. So these have to be 0. And you can verify that, right, by looking at the Punnett square. There are no offspring that will have red flowers. There are none that will have white. All of them are going to be pink. All right. So we're not, you can't do really Punnett squares for these, but I do want you to know that there are other traits that are influenced by more than a single gene. These are called polygenic traits or polygenic traits. Things like hair color, eye color, height, weight, and they can also be influenced by the environment, like if you go outside and you tan during the summer. Finally, I just want to end by mentioning epigenetics in that not all of who you are and what you look at like comes down to DNA. Sometimes your body has genes that it turns off because it's not beneficial for you to be using them. Sometimes it has genes that it turns on that weren't on in your parents, in one of your parents, so that because it's reading some sort of input that it would be beneficial to now be using that gene. So when genes are silenced, it's called gene silencing. Very nice. It's basically what happens is those chromatin fibers, the wound up hoses of DNA, they can open up and allow DNA to be accessed, or they can get closer together and prevent DNA from being accessed, which would be gene silencing. So they can put little tags on there, and chemically it will put the DNA closer together so it can't be transcribed, or spread it out so it can. So this is how your body turns on and off genes. So just because you have a particular gene doesn't necessarily mean you're going to express it, because your body has other mechanisms for regulating that. So the word epigenetics means on, above, or near. So it's like near genetic mechanisms. So there are modifications like adding little methyl or acetyl groups to parts of these like little nucleosomes or histones, and it makes the DNA either inaccessible or accessible. All right, so that's where we're going to stop. I know that was a lot today, but it all goes together and builds on itself. So I will link some stuff for practice problems if you want to use those.